Hello boys, welcome back to another session of video tutorials, and this session is meant for class 11th biology and the class 11th biology students, and this is meant for 28th January 2021. And as all of us know, we are into the revision mode and we are revising in the form of test paper 2. And in this session we are going to deal with chapter cell cycle and cell division. The main motto and the purpose of doing this revision in the form of a test paper is to make you familiarized with the type of questions, topology of the questions, and how to present your answers. Like, you may be knowing the answer, but how to present your answer is most important, so that you impress the evaluator. So as usual, we have got 1 mark questions, we have got 2 mark questions, we have got 3 mark questions, and most important, long answer type, we have got 5 marks question also in this chapter. Remember, in our previous session, biomolecules chapter, there were no 5 mark questions. But here you got 5 mark question. This gives a clue which are the chapters that you need to focus for long type questions. So keeping that in our mind at the background, here we go. Question 1. At which stage of meiosis crossing over of genetic material takes place? Crossing over. What is G0 phase? Name the cell division concerned with cancer, hereditary disease. Why is meiosis called as reductional division and mitosis is called as equational division? Write 3 processes that take place in interphase. Enumerate the significance of mitosis. Question 7. Write 6 differences between mitosis and meiosis. Very much expected question. And what are homologous chromosomes? Homologous, similar looking. What happens to homologous chromosomes during meiosis? And mega question, big question, expected to write something like 10 to 12 sentences, and this type of questions must be supported with beautiful sketches and proper labeling to enhance your answer. What is it? Question number 9. What is mitosis? Give brief account of mitosis in animal cell. That means you have to describe each and every substage of mitosis. Take a paper, start writing the answers, and once you finish it in next 30-35 minutes, come to the answer session that I am providing. Compare your answers with the answers that I am giving so that you know your strength and your weakness. Convert your weakness into strength and build up on your strength that you have already, so that you get good marks. So crossing over, of course, as we expected, it is pack 18, that is subphase. As all of you know, you may recall from your memory from the class 11th, regular video sessions, wherein I have told you that prophase 1 has got further 5 subphases. That is leptotin, zygotin, packaging, diplotin, and diakinesis. So it is the packaging in which the crossing over takes place. Question 2. Some cells in the adult animals do not appear to exhibit division, like heart cells, and many other cells divide only occasionally as needed to replace cells that have been lost because of injury or cell death. These cells that do not divide further exit G1 phase to enter an inactive stage called quiescent stage or G0 phase of the cell cycle. Cells in this stage remain metabolically active but no longer proliferate unless called on to do so, depending on the requirement of the organism. Question 3, of course, mitosis obviously. Question 4. In meiosis, the number of chromosomes is reduced to half. Like the mother cell will have diploid number of chromosomes, 2n. N is equal to number of sets of chromosomes, like 2 sets when we say 2n. Where in our case, n is equal to 23. 23 into 2, 46. So the leaving the gonadal cells, leaving the gametes, all others will be 2n. And this 2n cells undergoing meiosis will form daughter cells with the haploid number, that is only one set of chromosomes. That's why we call it as reductional division. And on the other hand, the mitosis, the number of chromosomes remain constant even after division. Hence it is called equational division or equal division. So mother cell also 2n and daughter cell also 2n. So the identity is maintained. Next, three processes are observed in interphase. What are those? Gap 1, G1 phase. It corresponds to interval between mitosis and initiation of DNA replication. During G1 phase of the cell is metabolically active and continuously grows but does not replicate DNA. It is only growth, cell grows. In a synthetic phase, S phase, very very important. This phase marks the period during which DNA synthesis or DNA replication takes place. This is a NEET exam question, UPSC, NDA question, and of course CBSE question. During this time, the amount of DNA per cell doubles. If the initial amount of DNA is donated as a 2C, then it increases to 4C because it doubles up. However, there is no increase in the chromosomal number. If the cell had diploid or 2n number of chromosomes at G1, even after S phase, the number of chromosomes remain the same, that is 2n. In animal cells, during the S phase, DNA replication begins in the nucleus and the centriole duplicates in the cytoplasm. So that is the importance of S phase. And G2 phase, gap 2, during G2 phase, proteins are, again, the neat question, proteins are synthesized in preparation for mitosis while cell growth continues. So how many phases? G1, S phase, G2 phase. Where is it? In interphase. Question 6. Mitosis results in the production of diploid daughter cells with identical genetic complements usually. The growth of multicellular organisms is due to mitosis. We should be thankful to mitosis. We are all growing. Cell growth results in disturbing the ratio between the nucleus and the cytoplasm. It therefore becomes essential for the cell to divide mitotically to restore nucleocytoplasmic ratio. Recent studies have shown that a cell becomes cancerous whenever there is a disturbance in the cytoplasm and nucleoplasm ratio. Thanks to mitosis, it avoids that disturbance. It tries to maintain that ratio so that cells become healthy. Next, a very significant contribution of mitosis is cell repair. How many times we are falling down during sports and games, accidents, we are losing so many skin cells, muscle cells, blood vessels, blood also, even bone, lot of fractures. But thanks to mitosis again, everything gets repaired within the specific days and specific time. So imagine our life without mitosis. Very, very horrible it would have been. So the cells of the upper layer of the epidermis, cells of the lining of the gut, blood cells are being constantly replaced. Mitotic divisions in the meristematic tissue, the apical, lateral cambium result in continuous growth of plants throughout the life. So plants also must be thankful to mitosis for their growth. Normal question, expected one, difference between mitosis and meiosis. Mitosis occurs in all the somatic cells, that is body cells. Somatic cells means body cells. Meiosis on the other hand occurs in gonads, reproductive cells, germ cells. Mitosis occurs both in sexually as well as asexually reproducing organisms, whereas meiosis occurs only in sexually reproducing organisms. The cell divides only once here. Here the whole process gets completed in two successive divisions, that is meiotic 1 and meiotic 2. Here synapsis is absent, in meiosis synapsis is very much present. No crossing over, no chiasma formation. Here crossing over takes place, chiasma also will be there. Thanks to this, because of the genetic exchange will be there. A cell produces two diploid cells. Here one mother cell produces 4 haploid cells. Going ahead, question 8. Homologous chromosomes are pairs of similar chromosomes having corresponding genes governing the same set of traits. During the heterotypic division of meiosis in leptotin, this is the first substage, chromosomes are thread-shaped and coiled. Next, during zygote, homologous chromosomes start pairing. Third, in pachytene, the chromosomes show thickening and shortening. Diplotin is marked by the cessation of attraction force between two homologous chromosomes. Uncoiling of homologous chromosome tends to separate them from each other, but remain attached at chiasma or chiasmata. Last stage, during diakinesis, the separation of homologous chromosomes is complete. Exchange of parts between chromatids of homologous chromosomes will take place. During metaphase, of course, metaphase is forgotten. During metaphase 1, there is lining of all the chromosomes in the center of the plate with the spindle fibers stretching them. During anaphase 1, the centromere of homologous compounds of bivalents repel each other, and after separation of the centromere, the homologous chromosomes start moving apart, north pole, south pole. In telophase 1, the chromosomes reach poles and become shortened. Question 9. Mitosis is an equational cell division in which the number of chromosomes in parent and progeny cell remains the same, 2n parent cell, daughter cell also 2n. And stasis, prophase, chromosome material condenses to form compact mitotic chromosomes. Chromosomes are seen to be composed of two chromatids attached together at the centromere. Initiation of an assembly of mitotic spindle, the microtubules, the protein components of the cell cytoplasm help in the process. Now, since it is a mega question, I told you your answer must be substantiated with the diagram, and most beautiful label diagram. Look at that. This early prophase, you can see the membrane. Since we are talking of animal, cell membrane will be there, nuclear membrane will be there, cytoplasm will be there, nucleoplasm will be there, chromatin reticulum will be there, and just chromatin reticulum starts appearing in the form of long thread chromosomes. In the late prophase, you can see nuclear membrane starts slowly disappearing, and long thread-like chromosomes become short and thick chromosomes. And in metaphase, as you are seeing, there is formation of spindle fibers, nuclear membrane disappeared, chromatophores are there, they are attached to these cytoplasmic fibers, and look at this. This is the characteristic feature of the metaphase. All chromosomes will be lying in the center. During anaphase, of course, the chromatid starts moving to the poles, north pole and south pole, one up, one down. This is the typical characteristic of anaphase. Spindle fibers will be there, nuclear membrane disappears, and the most important one is centromere splits, and chromatids separate, chromatids move to opposite poles. And coming to the last, telophase. In telophase, of course, the nuclear division is complete, karyokinesis is over, and formation of two nuclei, and there is a construction of wall, cytoplasmic wall, that means cytokinesis is also over. That results in the formation of two daughter cells, and the two daughter cells will be having reappearance of all the cell organs